Okay, so for the next part of this lecture, we're going to talk about, again, the binding of O2 to hemoglobin and myoglobin. So we need to define some terms first. And so we're going to discuss basically proteins that are ligand binding proteins. Now, proteins can bind different molecules, and so these molecules that they bind specifically to are known as ligands. And so let's say we have this protein, and we're going to call this over here, this is called the ligand binding site. Okay? Now, if we have a ligand that fits into this site, so this binding site is actually created by, again, the particular fold of the protein, and so at a certain spot within the protein, the amino acids surrounding this ligand binding site dictate sort of shape, a molecular shape that only certain molecules can bind into. And so we can think of, for instance, we can think of a couple different potential ligands that might fit in here, but we see that the shape of this blue triangle here is the only one that really matches. And so in this case, what we would see is the protein and the ligand would form an interaction, but only for the blue ligand, because it is, the shape of the active site is complementary and fits this ligand here. However, the circle ligand, for instance, doesn't have the correct molecular shape to fit into this ligand binding pocket, or this ligand binding site rather. And so it's not going to bind to this protein. And so that's actually a really important, that's really important for thinking about what proteins do in general. So proteins have evolved in order to have certain functions that are useful for life. But just as importantly, they also exclude other functions. And so one of the hallmarks of protein are that they're specific. So they don't just perform different chemistries, but they do it specifically. So in this case, the specificity is related to this protein binds a specific ligand, and in that case, that's this ligand, and it excludes this guy over here.